Okay. Hello, everyone. Good afternoon to colleagues here in Singapore, friends in Asia, and good morning to our colleagues and friends and partners in Europe. And I hope everyone is okay. Um, and thank you for being with us today. So, we are going to have our second part of this series of webinars, which we started last week. And this time we are going to talk about practical solutions for routine monitoring and validation requirements in the pharmaceutical or healthcare industries. Okay, just to give you an idea about the outline, we will be talking about common sterilization methods in pharma healthcare industries. This is moist heat or dry heat sterilization. We have ethylene oxide sterilization lined up, and we'll also be talking about hydrogen peroxide sterilization. Then a bit about process qualification, and this will be more into details in the next webinar session next week. And then we will also be talking about concepts, the validation concepts in pharma and healthcare, documentation and compliance, because pharma and healthcare is governed by standards and norms. So just like how we started last week, we would like to wish everyone and your families, all your loved ones, all the best and good health during this current COVID-19 situation. And I share with you a quote from Naval Ravisant: three things in life: your health, your mission, and the people you love, and that is it. Okay. Now, some of you may not know about Xylem yet. So, who is Xylem and what is Xylem? As Xylem word actually means the tissue in plants that brings water upward from the roots. And this is how we are trying to live our company as what it was spent, which is a living global water technology company. And yeah, we are world's leading water technology companies with over 5 billion US dollars from 2 years from 2017 revenue. And we started with 16,500 employees back then, represented in 150 countries in 7 continents. Um, this is just to illustrate that we are a global team, and we are creating advanced technology solutions to the world's water challenges. So, going from left to right, we have solutions for transport, treatment, dewatering, applied water solutions, and the group that I belong is in the measurement and control solutions, specifically the analytics business unit, and then that's where Ebro belongs as well. So you can see from here that Xylem provides this portfolio, and these are leading product brands and applications expertise from the people. My team, um, I think we make the products even perform better with our expertise. Xylem Analytics breakdown in terms of the global business, but it's only for Xylem Analytics. So we have process resulting to 30% of our revenue, the laboratory where Ebro belongs at 25%, um, the ocean at 10%, and the surface water at 35%. Okay, now we go to the main topics, but before doing that, we would like to share with you a quick brush up in one slide with what we talked about last week. Last week was about washer and disinfectors and sterilizations in the CSSD, which is the most important unit in hospitals or medical industries. Okay, so this is the workflow in the CSSD. Um, the bulk supply, which is in the general store, and the operating theaters and the patient rooms, which is represented in the wards and units department. And these are all collected for disassembly. So all supplies from the general stores and the wards and units are collected and received and disassembled. And those supplies, which are considered consumables and disposables like cotton, gauze, etc., are separated immediately. Then the instruments, the gloves, the rubber wares. Instruments, gloves, rubber wares, and the rest of the accessories are brought to the washer and disinfector unit. And this is where we have talked about our solutions, which is the wireless conductivity logger and the pressure or and temperature loggers. Then it goes to the assembly, then inspection. Inspection also includes the packaging and the labeling. Then they are placed in the pressed sterile storage or pressed sterile storage. They don't get to the sterilization process immediately because sterilization only comes when there is demand. And this is where our Ebro solutions comes in, um, which is most importantly the Bowie and Dick test, the EBI 16 logger. Then after sterilization process, and when they are successful, then they are brought to the sterile storage before distribution. So that was what we all talked about last week. Now our topic is about sterilization methods and processes in pharmaceuticals and healthcare industries. Um, just like in the CSSD, we have different cycles and procedures, but the same objectives, and that is reducing spores, create microbial inactivation, and ensure that commercial distribution of sterile goods are achieved. So the last one, which is quality without compromising on safety, this is the only objective not common to hospital operations. In hospital operations, they hardly look into quality because they are after absolute microbial destruction. But in pharmaceutical, they look at all the, aesthetics, the presentations of the medicines, the IV solutions, and the packaging. They need to put in consideration on quality as well. Um, why am I starting with or using this COVID-19 situation is because it is important for us to know about what we are doing with PPEs and how they are made safe and sterile to the users. Okay, And this is what we use for flattening the COVID-19 spread. Most of the PPEs, which is the personal protective equipment, but mainly we will be talking about um, face mask. Um, Under PPEs, during this COVID-19 um, spread, we are referring to coverall, goggles, face shields, gloves, head covers, and also the disposable shoe covers. Yeah, the most common PPE used by people is the face mask to filter aerosols. Aerosols, I use the term because it's referring to suspensions of tiny particles, and that's also the bacteria present in the air. We would like to categorize the different kinds or different types of mask. We have three actually. The surgical mask. Second is the N95 mask, and then we also have the elastomeric respiratory mask. I think the most common that we use right now is the surgical face mask. Um, it is recommended for people who are infected individuals, so they are not increasing the risk of transmitting the viruses to other people in the vicinity. It protects, but with some limitations because they are not so tight and they can only protect against large droplets of bodily fluids. Um, they are also prone to leakage around the edges, especially when breathing or inhaling. And lastly, the lower level of respiratory protection um, is not really blocking all the airborne particles. It must be considered as a single-use disposable mask. Yeah, there's no way you can reuse surgical face mask because they are just fabrics. Although non-woven for better bacterial filtration, they are made of plastic as well for the nose wiring or lining, so you have a good fit when you're wearing it. And the most important material is the polypropylene for better filtration efficiency. Um, so this type cannot be decontaminated, so it's really single use. Um, it is safe and reliable, but not all brands are. Um, it can be made sterile by dry heat sterilization, um, but you cannot decontaminate it when using high temperatures because it will damage the surgical face mask. So, how are people, how are the suppliers and manufacturers of these face masks make it safer for us is by ethylene oxide sterilization. Um, Ethylene oxide is a gas and it's explosive gas, and it undergoes four phases. That's preconditioning or conditioning, exposure, which is sterilization, exhausting for post conditioning, and purge, which is pulling the vacuum out of the chamber. And this takes long hours, many hours to complete. And there are four primary variables. Of course, the ethylene oxide concentration itself, but the parameters that needs to be measured are humidity and temperature and time. And that is why we have these loggers in this slide that is meant for only ethylene oxide chambers. Ethylene oxide is a low temperature gaseous process which is used for many different kinds of healthcare products which are sensitive to high temperature. Um, the use of vacuum-based process, ethylene oxide sterilization can efficiently penetrate surfaces of most medical products without destroying their physical properties. So it is safe to use for surgical face mask because materials are not exposed to high temperatures, moistures, and radiation. Um, but ethylene oxide processes requires regular validation. And I don't know one company with ETO chambers not validating their processes at all. So they have to validate the four phases and the four variables that we have talked about in the previous slide. And this is the preconditioning where we expose the mask to tropical environment for at least 12 hours, but stable at 55 degrees Celsius at 70% relative humidity. So this is the most critical phase of the ETO process. Then followed by the exposure, which is pulling the vacuum and exposed to the ETO gas, usually for 4 to 8 hours. It varies though to some products, but I'm just referring to um, surgical face mask of polypropylene materials in this example. Then the post conditioning, so it's like all air out and only ethylene oxide gas inside for 8 to 12 hours. So this is what we need to measure when we validate the complete ethylene oxide sterilization cycle. Moisture is very critical because it is used as a transfer agent for ethylene oxide to penetrate the products and the packaging. Because with ethylene oxide, products like face mask are not individually layered, they are already inside the packaging. Then the last cycle or phase is the purging, which is pulling a vacuum dramatically to dry the product. So therefore steam is increased to replace lost moisture in the product, and this influence of pressure is also a factor in helping ethylene oxide permeate the product. So pulling a vacuum pulls ethylene oxide into the product, as simple as that. So validation of ethylene oxide sterilization cycles. You know, Ebro, we have a good line of different temperature sensors and pressure sensors and humidity sensors. You can see here in the photos, and I'm sure Ebro Germany will slap uh, my back because these are the wrong luggers. We have to always check ATEX mark as represented with this one, the EX mark, which means that these three items must be used for ethylene oxide sterilization processes because they are explosion proof. You have to remember that. Other than face mask, ethylene oxide sterilization is used for other medical PPEs and devices as well. You can see here these are some surgical drapes, and the most common thing is these first aid kits like band-aids um, and other medical products. So we go to the second type of mask, which is the N95 mask. It is better than surgical face mask. The N95 respirator masks are sealed. They're more tight fitting, and it forces all air through a filter, which is designed to prevent more than 95% of zero point 0.3 microns of human droplets and particles. So um, this is what is recommended for infected people and also for the people wearing it just for protection. And reuse is possible, not before. Remember, not before. Just now, they can be reused. They can not be recycled as simple as just like reusing it after 2 3 days of wearing, um, because you need to disinfect the N95 mask. They can be sterilized by ethylene oxide as well. But the most effective sterilization method for this is the hydrogen peroxide. So contaminants and other viruses can be inactivated by hydrogen peroxide mask, just like how a steam sterilizer can reduce and inactivate microorganisms. So how they are made sterile? Um, yeah, as I mentioned, it can be made sterile with steam sterilization in autoclaves, but this is not a good practice because it's causing them damage because of the very high temperature and the pressure and very high pressure. Then ethylene oxide, um, manufacturers of N95 masks, they're using this, but it takes a very long time to complete a single process or single size cycle. It's like taking almost 2 days to finish one. So the most effective method is the hydrogen peroxide sterilization, and this is just perfect for the N95 respiratory mask. Um, we would like to explain the various type of masks, so bear with me because these are useful for infection control. And these different procedures that I'm mentioning, they are available to help reduce some of the risk involved with the reuse of masks, such as the N95 and the other respirators. So take this very seriously, guys. Okay, how hydrogen peroxide sterilization, um, because they don't cause any significant degradation to the respirators. They are recommended actually when validated with spores of Geobacillus stearothermophilus. This is the most common spore strips that are used to validate sterilization processes. Don't worry, this Geobacillus stearothermophilus is a surrogate microorganism. They're not natural, so they are very well handled. But just to mention that when using this with autoclave operations and also using the same spore strips of Geobacillus stearothermophilus in hydrogen peroxide, they both produce the same six log reduction, but they require validation. Okay, so N95 mask, they are under the regulated FFRs. FFRs means filtering face piece respirators. So N95 masks, as I mentioned a while ago, they are not approved for routine decontamination and reuse as a standard of healthcare. However, just days ago, due to shortages with N95 masks, FFR decontamination and reuse is now considered as a crisis capacity strategy. And you can see here the photo that I used in this slide. The regulatory authorities heard this plea for saving N95s for reuse. But just days ago, to be specific, just 2 days ago, the US is implementing this decontamination processes already. However, validation protocols are available since 2016. We go to details now with hydrogen peroxide sterilization. These are commonly known as the plasma sterilizers. They use a very low temperature um, and vacuum and commonly used to sterilize heat-sensitive devices and materials. So it involves filling the sterilizer chamber with hydrogen peroxide vapor. And once the sterilization cycle is complete, the vapor is vacuumed from the chamber and converted to water and oxygen. So this is typically how an H2O2 sterilizer works. They also use four um, primary stages. One is a pressure reduced to a very high vacuum. Then the liquid hydrogen peroxide is converted into vapor. Third, now under high vacuum, vapor fills the chamber and contacting all surfaces. And the last, after sterilization, the vapor is vacuumed from the chamber and converted into water and oxygen. So the entire H2O2 process is carried out in a constant temperature of 55 degrees Celsius for N95 masks. So this is how you are supposed to measure your H2O2 sterilization in order for you to get an indication whether you have achieved um, the correct degree of sterilization. So the challenge here in measuring this kind of process is how to measure pressure in a very low level down to as low as 0.01 kilopascal, which is equivalent to zero point 0.1 millibar. This is the most important parameter. As long as I'm concerned, this makes the whole process successful. Okay, the plasma phase, an electromagnetic field is created to produce low temperatures to cloud that contains ultraviolet lights or UV lights and free radicals. The vent phase must be measured because the chamber needs to have an equilibration between pressure and temperature. So this will enable the door to be opened safely. So there is no need for aeration or cooling down because the devices are ready for immediate use after completion of one cycle. So you must remember that applications for Ebro solutions for this is nearly very specific only for H2O2 sterilization. And that's because of the requirement of one logger to measure very low pressure. And we have a solution for that with the EBI 12 TP290 um, because it is very accurate as well, measuring at very low pressure. Now we go to the third type of mask, the elastomeric respirator mask. And this is most effective of the FFRs. And this is highly recommended for healthcare workers and people in the front line. Um, these are commonly available in half face piece, um, very tight fitting respirators, and they are made of synthetic or rubber materials, permitting them to be cleaned repeatedly and reused. So this is also the most expensive in the other compared to the other two types of mask. So cleaning and disinfection for elastomeric respirator mask are much easier because they are very robust in design. But then again, an SOP must still be followed because once again, when using elastomeric respirator mask, you are wearing them to deal um, with bacteria such as the viruses in the air and you are in an environment where infection is paramount. So the most common method to decontaminate and make this FFR safe for reuse is either by um, steam sterilization after a good disinfection and cleaning processes. The fourth, I'm sorry, this is a typo. This is the fourth mask recommended recently, just recently, by government agencies, the cloth face mask. Um, this is the least effective of the face mask, but this will help us cope up with the situation because we don't know for how long this will take or um, will be, in our environments, maybe in few months or maybe for next year. We don't know. So this is why we are resorting to this. But once again, disinfection, cleaning, and disinfection must still be observed. A cloth face mask, and there's a lot of homemade ones. They can provide very light protection, but it could help with reminding you not to touch your face. It also helps prevent the spread of germs from the person wearing it to others, but only for large droplets. So when people coughing and sneezing and wearing cloth mask, um, well, the risk of spreading um, the aerosols in the vicinity is reduced. So kindly inform all your customers about hydrogen peroxide and ethylene oxide sterilizers. It is very important because they are high in demand for keeping all the face mask and respirator masks safe uh, for people to wear. We have the solutions for all their validation and qualification requirements um, coming directly from Ebro. If they cannot purchase, considering the situation, um, we are really considering that maybe we could rent out the luggers or you can avail of the silent validation services. Um, I am I remain in touch with um, important people from the industry, um, thermal process authorities, consultants, um, healthcare experts, and I also know some very good people from Italy, like Roberto um, from Tektronic. We are we can help you with some validation guidelines for all your processes. So please call on us if you have some requirement for validation and requalification of these processes. So the question, are we still on with our session's topic for today? Because I have been talking about masks and how they're sterilized and disinfected. I could see that when you see the title of our session for today, it says practical solutions for your routine monitoring and validation requirements for pharma and healthcare. But the answer is yes. Other pharmaceutical and healthcare products, aside from FFRs, they are processed in the same manner and is always guided by standards and norms, and therefore performed with standard operating procedures and or validation protocols. So there's nothing different when dealing with masks and other pharmaceutical and healthcare products, as long as you're dealing with steam sterilizers, moist and dry heat sterilizers, hydrogen peroxide sterilizers or plasma sterilizers, and ethylene oxide sterilizers. They follow the same guidelines, and the protocols derived from these processes are almost exactly the same. Um, then I mentioned to you a while ago these partners and teams of consultants. They are just around available to help and assist you. We can provide you with thermal process validations, process adequacy determination and establishment, especially in the food and pharma, um, ethylene oxide and H2O2 validations. I'm beginning to train people um, performing and implementing validation procedures for these processes. And we can also provide trainings for all the above. But we would like to start this with our dealers and partners all over across the globe. We would like to make some review on validation and routine monitoring. Um, just very briefly, validation it's it's a cycle, it's a concept, and with the essentials coming from IQ, OQ, and PQ. I think from the previous webinars, I have been talking about IQ, OQ, and PQ, and I hope that you are able to understand these different stages of validation already. Validation, on definition, it's a complete presentation and verification of facts. Um, that's covering the processes, the machineries in your facilities. And the most important part of these validation stages are the validation reports, um, because this will present everything about the IQ, OQ, and PQ as a whole. Then routine control is more on periodic checks. Um, Ethylene oxide, hydrogen peroxide, they need periodic checks at the moment. So the frequency of performing these routine controls, though they rely heavily on management and leadership of your organizations, I'm pretty sure that the frequency for routine controls, especially when referring to ETO and H2O2, um, have changed. These processes, once again, when you're performing validation and routine monitoring, they must be conducted by authorized and knowledgeable personnel. Now we go to IQ, OQ, PQ according to their definitions, but you will have a copy of this presentation and I will not go through all this. And this is important, the standards and the norms. Um, the reason why we cannot get away with observing the procedures, including the validations of sterilizers, is because we have existing norms. And these norms, I'm pretty sure they are observed worldwide, whether it's European or American. ISO 17665, we observe it from here, we follow it from the Philippines, um, in the US and in Europe, and it's a very good standard to read and be aware of. Then you know that validation and routine control is necessary. So ISO 11135 or the 11135 for ethylene oxide sterilization. Um, this is very specific, um, referring to procedures because once again, handling and operating ethylene oxide sterilizers are, let's say, close to dangerous. Um, so therefore, all these programs being put in for their cycles must be validated and measured all the time. Although with validation, They are like regulated and only um, requires the operators for periodic validation procedures. Um, you must be able to provide, I mean, the dealers must be able to provide a ready solution whenever these requirements come out. Long ago, there were no regulations for hydrogen peroxide, but now we have the ISO 14937 specifically for low temperature sterilization standards. And this is a good read. I find it a very good read. So I'm recommending that um, you go and browse through this standard when you have the time. And in the pharma and healthcare, we also have different standards and norms for oven and incubators. We have talked about this last session, but the ISO 17665, when it comes to sterilization, is the most common standard that are observed across the globe. When we are doing all these thermometric measurements, the only thing that matters is temperature, pressure. With the ETO, it's relative humidity, time, but these are all like data points and will not mean anything until they are evaluated properly. And we have the softwares, and I recommend the WinLog validation for a thorough and more accurate evaluation of your sterilization processes. So it's very important um, that we know that all those stages in the sterilization processes and cycles are met and their objectives are achieved. So this is just procedures, an example of an SOP, and the recommended data loggers for all those processes that we have talked about. Um, we don't have too many competition for this special logger. So please check your catalogs and check about this logger that can measure um, down to zero point 0.1 millibar. So lastly, now going back to software that Ebro provides us, the WinLog Pro and the WinLog validation. There's one request from yours truly, and that is we kindly, I am asking you to learn the software. The only way we can all be successful with all these promotions about solutions for validations is for you yourself to run the software confidently by yourself. So I'm planning of holding webinars for only for end users and dealers, and I'll work this out with Michael and Rumiko and also with Ivan in Ebro, Germany. Um, it should be nice if you are able, I mean, all of you are able to produce and generate validation processes and validation reports as this one. And that can only be made possible when you are serious with learning the software. So we are running out of time. Um, that's it for today. Um, Next webinar session, we will be talking in detail about IQ, OQ, PQ, and the validation um, solutions that Ebro can provide. So thank you again, everyone. If you have any questions, then now is the right time. If I may please ask Michael to join me now here.
Hello, Allan. Great
Hi,
presentation
Michael.
as always.
Thanks, Michael. I hope so.
Before we start with the questions, I just wanted to ask, was that you behind the Darth Vader mask?
Yeah, it is. Yeah, it is.
Great. Well, as Allan mentioned, now is the time for you to ask questions, and feel free to type your question into the question chat box that's on your screen. We'll go through them one by one with the time that we have remaining. Any questions that we can't answer within the time period, these will be answered directly by Allan via email. Using the GoToWebinar program, we are able to connect the email address of each attendee with the question that they're asking on the system. So no worries, we will get back to you.
Thank you
So
for
again,
mentioning that, Michael. I promise the questions from the previous webinar session, um, I will, I promise that I'll answer all of them not later than Thursday this week.
thank you, Allan. We know you're a busy guy. Okay, well, we have quite a few questions rolling in now, so let's get started. The first one, if you use EBI 12, for ethylene oxide sterilization process, do we have to use the silicone protection EBI 12 since the chamber of ethylene oxide sterilizer is wet?
Um, Now, will not really recommend it. You can, of course, use the silicone protection, but the ethylene oxide, it's bigger than my house sometimes. You know, they can just position the loggers that you are going to use in a safe area, and it is an EX approved logger, so you don't need any protection um, for the loggers when using in ethylene oxide sterilizers. But just just be sure you're using EX type loggers.
Great, thanks, Alan. Let's move on to the next question. Is there a certain volume or volume ratio of hydrogen peroxide volume to sterilizer chamber volume?
Yeah, yes, that is a very a good question. It's depending on the dimensions of the H2O2 chamber or sterilizer, and the configuration or your loading configuration. You need to perform an IQ-OQ before you use the H2O2 um, sterilizers in actual use. So then from performing this test, then you are able to perform or result or establish the correct volume ratio or volume to volume ratio.
Thank you, Alan. Next question. Uh, which kind of parameters are important in ethylene oxide and hydrogen peroxide sterilization?
For ethylene oxide sterilizers, the most common that people are using is the temperature loggers and the RH or humidity loggers. But we also recommend using the pressure loggers, although in some protocols, it's not requiring the use of pressure loggers anymore, because what is important with ethylene oxide is the correct control of the moisture represented by relative humidity and the temperature setting. Now going to H2O2, it's precisely why I have emphasized this low pressure logger, because it is the most important logger that needs to be used in H2O2 processes combined with the temperature EBI 12 loggers. Thank you, Michael.
Thank you, Allan. This one's, I'm not sure if I'll catch this. Perhaps you will, Allan. For the WinLog software, is this 21 CFR with audit trail? Is that a full 21 CFR?
Yeah, it's the FDA 21 CFR Part 11. What about it?
They're asking if it has an audit trail.
Oh, yes. Yes. Not all versions, though. We have the WinLog Pro, and it is compliant to 21 CFR Part 11. And of course, the WinLog validation, 100% compliant to this FDA 21 CFR Part 11 as well. It has audit trails, and you can also include your electronic signatures when generating reports.
Excellent. What is the fundamental difference between validation and calibration?
Calibration. Calibration is just a verification of what is specified by the manufacturer as to what as the accuracy of a device. So calibration is merely that it gives you um, the correct indications of uncertainty and accuracy um, in one procedure, while validation is a verification not only of a machine but how they perform. Validation is verifying that the process is resulting to how they are supposed to perform. In terms of sterilizers, validation is from the very start that you switch on the sterilizer and to the very last point that it gives you a report that would show that microbial inactivation as represented by the lethality values are presented. So it's very, very different, these two things. I can spend a lot of time discussing about calibration, but I don't think now is the right time. There's a lot of misconception about calibration. First and most common is that when you calibrate, then you automatically adjust the accuracy, but it's not. But anyway, Michael, if we can discuss about that later on after the show.
Sounds like a good idea, Allan. All right, and going back to the WinLog Pro software, can this be used for reporting on sterilization?
Oh, yes, 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 it can. But you need a lot of um, integration of the different stages. But yes, it can be performed. When I started with Xylem, I was very comfortable with WinLog Pro, and I have used it for validation. But when I learned about the WinLog validation, man, it's far far better than the WinLog Pro. But the answer is yes, Michael.
Great, thanks, Allan. How many points are needed for the validation of ethylene oxide and hydrogen peroxide?
How many points depends again on the dimension of your chambers. The ethylene oxide normally they require up to 12 measuring points, but then again, it must be representative of the whole interior chamber, the vessel or the chamber. So it's very, very dependent on the dimension. And the same is true with H2O2. But H2O2 is very small. Most of them are small. So a temperature measuring point of 4 initially and 1 pressure can do the job for a complete profile.
And Allan, just to confirm, is that using a data logger?
Yes, that's using a data logger, but I'm trying to be specific on the measuring points because one data logger you can have as many as 4 measuring points in one logger. So yes, I'm referring to the data loggers, but the measuring points as well.
Okay, great. There seems to be a lot of interest in the WinLog software. There's another question here. Do we have any demo WinLog software that our users can get trained on?
You know, management have already caught my attention with giving free softwares. So please, when you want to learn the software, learning most of the time they're not for free. I'm sorry to mention that. So you have to invest on your own demo, initial starter kit, you know. Um, it will help you, I'm very sure. Um, if I give you a software, you cannot hardly do anything with it without the loggers. So please buy a starter kit, the data loggers that comes with the software. Thank you, Michael.
Thanks, Alan. Okay, next question in. Do these sterilization procedures, the ethylene oxide and hydrogen peroxide, do they impart some kind of odor onto the sterilized product?
Um, yeah, you know, in before you can ever perform any validations in an ethylene oxide, you need to submit a validation protocol and make your QMS or quality management system or validation managers to approve them. So there's no shortcut. First, plan. Second, write the validation protocol. And third, get the green light to perform or implement the procedures. Did I answer your question, Michael?
Well, it sounds like it. It's not really my question, so
Oh, sorry. Okay,
but
come
that's
again.
fine. Yeah. And
Yeah.
again, to our audience, if there's any questions that were not answered uh, to your understanding, then please contact Alan directly, and I'm sure he'll be happy to help you out.
I'm
Um,
sorry about that last question, Michael. Can we
no,
have some?
that's fine. No, it sounds clear to me. All right, and there are a number of questions about your follow-up webinars, Alan. And just to share with everybody, there is a follow-up email that will be sent to you automatically through from GoToWebinar, which will include a link to the recording of this webinar, so you can view it again at your own convenience. And in that email is also a link to our website, which has a list of follow-up webinars that Alan will be presenting next week and the week after, as well as additional webinars that are being hosted by other guest speakers from Xylem Analytics. So feel free to drop by the site, browse through our available webinars, and feel free to register at your own convenience. So Alan, unfortunately, that's all the time we have today. Would you like to say goodbye to our audience?
I just want to share what myself and Rumiko were talking about a while ago. We are all working from home. I think most of us are. So our health is very important. So Rumiko and myself were talking about healthcare reforms, and I'd like to share it with you. We just found out that the most real and most authentic healthcare reform starts in our kitchen and not in the World Health Organization. So please stay healthy, everyone. Make the most of the quarantine time with your family and still be productive. I'm looking forward to meeting you all again next session. Thanks everyone.
Thanks, Alan. Goodbye, everyone. Stay safe.